So now we'll be looking at covalent catalysis. So covalent catalysis is another important strategy which is employed by many enzymes to catalyze their chemical reaction. So the basic feature of this covalent catalysis is that a transient covalent intermediate is formed between the enzyme and the substrate. So for example, if we consider the hydrolysis of peptide bond, so water attacks the peptide bond to break it. So without the presence of enzyme, for example chymotrypsin, this bond breakage is very slow and it takes years to digest the peptide bond in absence of the enzyme. However, chymotrypsin possesses a powerful nucleophile at its active site, which forms a transient covalent bond with one of the reaction intermediates. So if we shall look at the mechanism, chymotrypsin is one of the enzyme which also is categorized under the general acid base catalysis. So here a base like histidine 57 generates the powerful nucleophile with serine 195. So it abstracts this proton from serine 195 to make it a powerful nucleophile which attacks this peptide bond. So this bond, this is the peptide bond. So the peptide bond is broken in the first step, and this is the covalent bond we are talking about, covalent. So however, this bond is not permanent, it is a transient step during the catalysis, and this covalent bond is broken by addition of water. So that's why it is called hydrolysis reaction. So this water breaks this bond between serine 195 and the half of the peptide to regenerate the active site of the chymotrypsin. So this is one of the very beautiful example, the chymotrypsin, which can be exemplified both in case of acid base catalysis and covalent catalysis. So to look at which groups or which amino acid side chains act as common nucleophiles, the aspartic acid, glutamic acid, histidine, lysine, cysteine, tyrosine and serine, which are rich in electronegative groups in their deprotonated state, can act as nucleophiles. And some of the common electrophiles include the carbonyl group, proton, the peptide bond, or the Schiff base, and the phosphate group. So these these groups are actually deficient in electron, so they can pull the electrons from the nearby electronegative groups. So we'll be studying more about this type of covalent catalysis in the subsequent videos.